Hello, here again with the same topic, Unit 4, Grade 12, Biology Evolution, of course. Yeah, continued. We have been dealing about one type of chromosomal mutation, which is the structural change, chromosomal mutation type 1, which is also known as the structural chromosomal mutation, also known as structural aberration. Structural aberration, chromosomal aberration, we can say. And therefore, we have seen to refresh your memories from these structural changes. We started discussing about deletion. Then we came to deal about inversion. Third, we stopped from translocation. We have seen this part, of course, translocation, the third type of structural change, translocation. We have seen this. It happens when segment of chromosome breaks off and then relocates to a completely different chromosome, of course. And we have also seen that this creates one part from one chromosome breaks and fuses. Also another part, another chromosome breaks and the two fuse. Therefore, it forms fusion chromosomes as one type of structural change. We have seen also these are two types, reciprocal translocation and Robertsonian translocation. Now, we shall see the first reciprocal translocation. What does it mean? This occurs when two different chromosomes exchange segments, and for example, a piece of chromosome 1 may break off and attach to chromosome 2, while a piece of chromosome 2 attached to chromosome 1. They are similar to the crossing over event, except that this may not be, of course, yes, this may not be equivalent exchange of segments. One chromosome may give larger parts while the other smaller parts. It's not an equivalent exchange. In some cases, it could be equivalent exchange, so that both chromosomes are equally exchanging similar portion of their DNAs. Therefore, it could be, of course, it is balanced or unbalanced. We shall come to the balanced and unbalanced type of translocation. We call it balanced if one chromosome is getting smaller portion so that it has part of that chromosome, genes of that chromosome, it may lose. The other gets a larger proportion, surplus of chromosome and portion of DNA or genes. This case, it is unbalanced. Otherwise, if the same length of chromosomes are reciprocated between two non-homologous chromosomes, it is balanced. Anyway, reciprocal translocation involves the exchange of pieces of segments of DNA, chromosomes, between non-homologous chromosomes. You can see here now this diagram tells you these two chromosomes are not homologous chromosomes. Now this one breaks from this portion and given to this one with the gene CDE, and this one breaks MN and gives the two genes. You see in this case this one is getting CDE gene from this chromosome, and this one is getting MN containing genes. Therefore, one gene above that one is receiving 3, the other is receiving 1. This may be considered as unbalanced. Otherwise, it is reciprocal translocation, of course. The other is Robertsonian translocation. Very important, very important. Robertsonian translocation, short ROP, is a type of chromosomal abnormality that occurs when the long arms of two acrocentric chromosomes, non-homologous acrocentric chromosomes, fuse together. The long arms, they call the long arms the Q arms below the centromere. I can show you what are acrocentric chromosomes. Let's ask, what are acrocentric chromosomes? Acrocentric chromosomes are those with very short PR, the upper arm, the region of the chromosome above the centromere. And in humans, the acrocentric chromosomes involved in this type of translocation are chromosome 13, 14, 15, 21, and 22. These chromosomes in human are the acrocentric chromosomes. So they are involved in Robertsonian translocation. We shall see one by one. You can see here diagram of chromosomes and classification of chromosomes based on the centromer. You see the centromer is near to this is the centromer is here, so that the portions above the centromer, which are known as p arms, are very short, very short. Of course, the one, this is telocentric, it has totally no p arm here. And the acrocentric chromosomes will have very petite or very small p arms and very long q arms. And such chromosomes come together and cut their q arm from the centromeric position usually and exchange. When they do this, we call this translocation Robertsonian translocation. Okay, therefore, don't forget, remember, acrocentric chromosomes are chromosomes with very, very small PR and very, very long QR. Such chromosomes come together, they cut their Q arms and exchange. Of course, they also exchange their P arms. When the P arms from two non-homologous chromosomes are joined, they are very minute, very small. Two very small parts create two very small components of two chromosomes come together. Still, they are very small, so that they will not stay in the nucleus. They go out from the nucleus through the nuclear pore, and when they get to the cytoplasm, they end up in lysosomes to be broken down, of course. Therefore, they are lost. Whereas the Q arms are very large, so that when they fuse and they form fusion chromosome, that fusion chromosome is very long. This fusion results in a single chromosome with two long arms and typically leads to a reduction in the total number of chromosomes in the affected individual. Yes, these individuals lose the two smaller portions, but they have two long arms formed by fusion of two non-homologous chromosomes. Example, let me give you example. Robertsonian translocation involving chromosome number 13 and number 4, which is relatively common. Plus the second example that I'm going to give you is Robertsonian translocation involving chromosome 14 and 21. You see? Now, Robertsonian translocation involving chromosome 13 and 14. You can see the short arm, you can see the long arm. You can see here two chromosome number 13 pairs. In pairs, we do have all our chromosomes in pairs, one from mom, one from dad. When these chromosomes undergo translocation during meiosis, you can see that the two pieces are lost. They are joined or they are individually lost from the nucleus. And this one 14, this one 13, these are translocation chromosome. Chromosome 13 translocated, on which chromosome 14 has been translocated. You see? Then what happens if an individual baby receives chromosome 13 normal from dad, chromosome 13 normal from mom, but when that baby receives one normal chromosome 14 from dad, but one translocated chromosome 14, which contains the long arm of chromosome 13, this is considered as one extra chromosome number 13. This one is considered as full chromosome because it is the longest portion, the longer portion, the Q arm. Therefore, you consider this case trisomy 13. That means chromosome number 13 is found in 3 copies because this is considered as one copy. Trisomy 13 causes what? Miscarriage. A baby with trisomy 13 in this translocated form, the fetus or the embryo the embryo will not continue, either miscarriage, spontaneous abortion. But in some babies, still scientists do not didn't decipher so far why some babies undergo miscarriage or spontaneous abortion, and why some survive and are born healthy. Of course, healthy means they do have many complications, which is known as Patau syndrome. Go and read the symptoms of Patau syndrome. Many symptoms, including even there are some underdeveloped nervous system. The second case is chromosome 14. You see here, chromosome 14. It could be chromosome 14 normal, chromosome 13 normal. Chromosome 13 is given together with the translocated chromosome 40. So in these individuals, chromosome 40 is found in 3 copies. So it is known as trisomy 14. This one is restless. This one, trisomy 14, totally do not allow survival of babies, and it causes miscarriage totally. You see, therefore these are two types of Robertsonian translocation involving chromosome 13 and 14. Now, the second example, Robertsonian translocation involving chromosome 40 and 21. Chromosome 40 and 21. Okay. Here, you can see when they are undergoing translocation, the two pieces are lost, and you have this one normal chromosome 40, one chromosome 14 with translocated 21 chromosome, one normal 21. Now, when babies receive two normal from dad, from mom, 21 chromosome, but chromosome 40, one normal from mom and one normal, one abnormal, that means translocated 21, containing translocated 21 chromosome from dad, this individual is considered as trisomy 21. It is considered as having 3 copies of one type of chromosome, what is 21 chromosome. So it is known as Down syndrome. These babies are born, but with many complications, including very low IQ. The other is chromosome 14 and 21. 21 here, both the chromosome 14 are normal from dad and mom. In this case, both chromosome 21, one obtained from one parent is 21, from the other parent is the chromosome 21 that contains the translocate chromosome 40. These individuals are considered as trisomy 40, similar to the case we have seen earlier. Trisomy 14 is total miscarriage, do not allow survival of the baby. You see, these are trisomies that happen because of translocation. Which translocation? Translocation that involves what? Acrocentric chromosomes. What are acrocentric chromosomes? These are chromosomes which do have very short P arm and very long Q arm. And they cut and exchange the Q arms, so that forming fusion chromosome, which is very bigger. Balanced. The other case of discussing translocations is whether the translocation is balanced or unbalanced. Translocations can be further be categorized into two based on their effects on genetic material. Balanced translocations and unbalanced translocations, of course. Balanced translocations, no genetic material is lost or gained, and where the chromosome is fully functional, and the individual may not show any symptoms. You see, this is balanced. Unbalanced, this results in extra or missing genetic material, and thus can't function as normal, which can lead to developmental disorders or diseases, such as Down syndrome. It can cause, or certain types of cancer, such as leukemia or blood cancer, can be caused by such type of unbalanced translocations. And you see, balanced because of the translocation in an individual, there is no missing gene. It contains all the genes intact. Unbalanced, the individual is receiving missing genes, lost genes, or even extra genes. In this case, having extra genes because of translocation or missing genes because of translocation, both are not normal. The last type of structural chromosomal mutation is duplication. Duplication, also known as amplification. An extra copy of a segment of chromosome is present within the chromosome, sometimes also called partial trisomy. They call it partial trisomy because part is found twice or thrice. In this case, this is a normal chromosome. Here, A, B, C on the P arm, D, E, F on the Q arm. When you go to here, A, B, B, C, the portion of the gene in B is repeated twice. So it is duplication. You may make it even A, B, 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 C, or A, B, C, C, C. You can repeat, or A, B, B, even you can see. Therefore, when part of a chromosome is found twice or more. And last, this is the summary of structural chromosomal mutations, mutation types. The diagram shows the first one, duplication. We have seen it earlier. Part of chromosome is repeated. Deletion, part of chromosome is broken and missing. You have to remember deletion is divided into two, terminal deletion and centric deletion or interstitial deletion, when it is from the middle portion breaking at 2 points lost. And also inversion could be pericentric and paracentric. Paracentric when it is not involving the centroma. Pericentric when it is not involving the centroma. The other is translocation. Translocation can also be divided into two: the reciprocal translocation and Robertson translocation. We have seen that. Thank you very much, and I will come by the last. I think the last part of mutation. The last part mutation. Thank you very much. God bless you. Bye bye.